In our body, our cells need energy to work properly and to fulfill their functions. This energy comes from organic molecules like glucose, lipids, etc., present in food and used under the form of ATP, adenine triphosphate, thanks to the mechanism named metabolism. Metabolism is composed of catabolism, degradation, and anabolism, production. The formation of ATP from organic molecules needs many mechanisms interacting together. Glycolysis from glucose, Krebs cycle, intermediate, oxidative phosphorylation, formation of ATP. I'll show you a drawing of it to well understand it. Glucose is transformed into glucose 6-phosphate with 1 ATP and then into fructose 1,6-bisphosphate, which form glyceraldehyde 3-phosphate and dihydroacetone phosphate. They make a reversible reaction between them and then form 2 phosphoenol pyruvate and finally 2 pyruvates. Pyruvates can be transformed into acetyl-CoA, into Krebs cycle, into lactate or ethanol.
And one more question about it. What is the regulation of glycolysis?
The most important is the regulation by exokinase in hepatocyte and beta cell of pancreas. It has a high affinity for glucose. The exokinase 4 is the glucokinase and have an important hyperglycemic role. Its affinity for glucose is more important than the hexokinase. During a meal, glucokinase is acting.
If you want energy, you must travel to the downest place of the cell, known as mitochondria. There's a lot of space there, and there's also 2 huge walls.
How are these walls?
It's a set of proteins and organic molecules. The fountain of energy is behind the wall. You must take advantage at the moment when the ion H+ travels from the matrix of the mitochondria to the space between those 2 walls. Try to enter in the matrix. Remember, in the matrix, there's a lot of energy.
But I don't understand what happened when the ions H+ are pumped.
In the intermediate space, there is a higher concentration of H+, and in the matrix, there's a lower concentration. A proton gradient is created. It's also known as electrochemical gradient. This gradient is a form of stored energy that can be used to generate ATP.
Is ATP the popular source of energy?
Yes, of course. It is capable to synthesize our DNA in addition to other essential functions.
What I need to this mission to enter in the cell?
Glucose. You need glucose. Once you have pyruvate, you will be available to enter in the mitochondrial matrix. This process is really important because it spends ATP and NAD+.
So does the pyruvate be my goal in this mission?
No, pyruvate is oxidized due to coenzyme A and become acetyl-CoA.
My grandfather told me something related about the consequence of this.
Yeah, sure. After all of this, Krebs cycles takes place. I have a question. What is Krebs cycle?
Krebs cycle or citric acid cycle is a central driver of cellular respiration.
Where does this happen?
This takes place in the matrix of the mitochondria.
Oh, but how does this happen?
Krebs cycle it takes acetyl-CoA, produced by the oxidation of pyruvate and originally derived from glucose, as its starting material and, in a series of redox reactions, harvests much of the bond energy in the form of NADH, FADH2, and ATP molecules. The reduced electron carry NADH and FADH2 generate in the CAC cycle will pass their electron into the electron transport chain and through oxidative phosphorylation will generate most of the ATP produced in cellular respiration.
How many steps are present in the cycle then?
The cycle includes 8 major steps.
And how many products are they?
2 carbon enter from acetyl-CoA and 2 molecules of carbon dioxide are released. 3 molecules of NADH and 1 of FADH2 are generated and 1 molecule of ATP or GTP is produced.
Okay. What will happen in oxidative phosphorylation?
You must enter in the electron chain, and you must travel through it, where the redox reaction occurs.
Redox reaction? What does this mean?
The energy released in the reaction is captured as a proton gradient, which is used to form ATP in a process called chemiosmosis.
So when I grab the ATP, can I get out?
Yes, because the flow of ions passes through the ATP synthesis, which takes advantage of the flow of protons to synthesize ATP.
I'm not sure. I have a lot of doubts.
Don't worry, you need help from other teacher. Go to talk with them.
Okay, I will.
Hello, welcome to my office. You can ask me one question and I will answer it. What do you want to know?
I wanted to know why is NADH better than FADH2?
NADH is very good at donating electrons in redox reaction. That's it, its electrons are at a high energy level, so it can transfer its electron directly to complex 1, turning back into NADH. As electrons move through complex 1, in a series of redox reaction energy is released, and the complex uses this energy to pump protons from the matrix into the intermembrane space. For further information, you must speak with another teacher. Good luck.
Thank you.
Thank you for coming. Sorry, I'm very busy, so you can only ask me one question.
Okay, how would the complex of transport chains work?
Beyond the first two complexes, electrons from NADH and FADH2 travel exactly the same route. Both complex 1 and complex 2 pass their electron to a small mobile electron carrier called ubiquinone, which is reduced from QH2 and travels through the membrane, delivering the electron to complex 3. As electrons move through complex 3, more H+ ions are pumped across the membrane, and the electrons are ultimately delivered to another mobile carrier called cytochrome C, CTC. CTC carries the electron to complex 4, where a final batch of H+ ions is pumped across the membrane. Complex 4 passes the electron to O2, which splits into 2 oxygen atoms and a set proton from the matrix to form water. 4 electrons are required to reduce H molecule of O2, and water molecules are forming in the process.
Okay, thank you.
I was waiting for you. Ask me whatever you want.
What does the electron transport chain do for the cell?
Regenerates electron carrier, NADH and FADH2, pass the electron to the electron transport chain, turning back into NAD plus and FAD. This is important because the oxidized form of these electron carriers are used in the glycolysis and the citric acid cycle and must be available to keep this process running.